The polymerase chain reaction, PCR, is widely used as a research technique in the field of biotechnology. The purpose of the polymerase chain reaction entails primer-mediated enzymatic amplification of specific target sequences in genomic DNA, resulting in the exponential increase of target DNA copies. The PCR is considered primarily a method for producing copies of a specific sequence. It is also a very powerful and precise way of altering a particular template sequence. Once the specific sequence is amplified, the DNA produced by the PCR can be used in many laboratory and clinical procedures and techniques. Every day, PCR is used to identify and diagnose diseases, solve crimes through analysis of genetic fingerprints for DNA profiling and amplifying ancient DNA. Only a few listed here, however, there are many more uses. The list goes on. The popularity of the PCR is primarily due to its apparent simplicity and high probability of success. The invention of polymerase chain reaction in 1983 is mostly credited to Kary Mullis. Also recognized was his technician who worked alongside him. At the time, Mullis was working for Cedars Corporation, and this company was one of the first biotechnology companies. One Friday night in April 1983, Mullis discovered a method of replicating DNA by amplifying any region of DNA by repeating cycles of duplication, which is driven by DNA polymerase. The reaction is easy to execute. It requires no more than a test tube, a few simple reagents, and a source of heat. Initially, the PCR used the Klenau fragment of E. coli DNA polymerase I to extend the annealed primers. This enzyme was inactivated by the high temperature required to separate the two DNA strands at the outset of each PCR cycle. Consequently, fresh enzyme had to be added during every cycle. The introduction of the thermostable DNA polymerase, TAQ polymerase, isolated from Thermus aquaticus, transformed the PCR into a simple and robust reaction which could now be automated by a thermal cycling device. The Klenau enzyme PCR was not a very specific reaction. The increase in the specificity of the TAQ PCR results in an improved yield of the amplified target fragment by reducing the competition by non-target products for enzyme and primers. PCR technology has come very far since its invention in 1983 and continues to develop and has potential for further improvement.
So from here, we're going to talk about how the PCR amplification process works on more of a specific and molecular level. So before we start the PCR reaction, we need a solution containing 4 specific ingredients. Firstly, we need the DNA that we want to amplify and replicate. Secondly, we need a special form of polymerase known as Taq polymerase, and we'll discuss why we need that specific polymerase. We also need the oligonucleotide primers, because these will allow the Taq polymerase to bind and begin elongation. And finally, we also need these dinucleoside triphosphates in order to supply the building blocks for elongation. So before we talk about the manipulation of temperature according to the specific DNA strand, we're going to talk about the structure of DNA itself and what's going to cause these changes. So we have the difference in bases mainly, but we're going to talk about the bonds or the strength of the bonds that occur as a result of the base-base pairings. So on the right here, I've just got the purines, which are going to be guanine, the red, and adenine, the green, and the pyrimidines, which are cytosine, blue, and thymine, yellow. Now, there are 3 key bonds that exist in DNA. One of them is going to be important in discussing how it affects the supplied temperature and the changes to the temperature, and that's going to be the hydrogen bonds here. So the hydrogen bonds occur between all 4 of these bases. So between guanine and cytosine, we have 3 hydrogen bonds, as shown here. And between adenine and thymine, we only have 2. So as you can guess, the more cytosine-guanine pairings that we have, the more energy, so the more heat we will need to add to break the DNA structure apart. And vice versa, if we have more of the adenine-thymine pairings, we will need less temperature. And quickly, we have the phosphodiester bonds, which occur between the phosphate groups and the ribose sugars. So here on the 5' carbon of this ribose sugar and this 3' carbon. And we also have glycosidic bonds, which are the pairings between the ribose sugars and the relevant base, as shown here. So the first step in the polymerase chain reaction that we have to do is denaturation, and we do that by increasing heat. Now we could do it by mechanical shearing force or by altering the pH, but manipulating the temperature is a lot more consistent and more easily replicated. So in just talking about hydrogen bonds, I've put in bold here the cytosine guanine base pairings, and in a thinner black line, these are the adenine and thymine base pair connections. So at room temperature, as shown here, the DNA structure is intact, but for step 1, depending on the number of cytosine guanine pairings and the number of adenine thymine connections, that's going to manipulate this temperature here. So the more blue to red connections, that will raise the temperature. But if we have more adenine and thymine connections, that will actually decrease the temperature needed to denature the protein. So, assuming that we've calibrated the machine correctly, what will happen is these bonds will no longer connect. So the two DNA strands will split apart so long as that temperature stays the same. So we've just finished denaturing the DNA strands, but now we need the oligonucleotide primers to join to the complementary base sequence in the flanking position in order to signal Taq polymerase for elongation. So we can't leave the temperature at 95 degrees, otherwise these primers simply won't be able to hold the hydrogen bonds between the bases. So we bring the temperature down to around 55 degrees Celsius. And I've also highlighted in the background that there's a high saturation of primers, because without the saturation, the original DNA strand simply joins back together. So it competitively beats the original strand from joining, and they link onto the flanking sequence, and the base is complementary to them. So I'll quickly draw in the complementary sequence. Now, they don't just join in any old fashion. It's very specific, and so the 5' end of this primer joins to the 3' end of the DNA strand, and vice versa. So the primers have joined to the complementary DNA sequence, but elongation won't occur without the presence and activation, I should say, of Taq polymerase. And the way we do that is by increasing the temperature from around 55 degrees Celsius up to 75 degrees Celsius. And the reason we do that is because Taq polymerase is more active in a temperature closer to 75 degrees. So it will bind to the oligonucleotide primer, and using the DNTPs, so the dinucleoside triphosphates that are within the solution, it will synthesize and elongate the complementary sequence of the mother strand. And that synthesis won't stop until we reach the very end, so I've shown them as a star symbol, unless a specific mutation within the strand or an endonuclease is present. So denaturation, annealing, and elongation was really only one cycle of the PCR process, and that yielded 2 strands. So the idea of this process is to actually yield billions and billions of strands. So after step 2, we have 4 strands, and as we can see here, this is the mother strand here, and this is the daughter strand here. So each time they denature, there is a new strand being built complementary to its own. And similarly, we could say that this strand here is this one here, and that this strand here is actually that strand there. So we're essentially multiplying the number of DNA structures by 2 with each cycle. So it isn't until we get to the 3rd cycle that we really get the target DNA that we want. So we finally got the one that we want, but we need billions and billions of these. So we actually end up by repeating this process, the final target DNAs, by 25, 30, but it ultimately depends on how efficiently and how accurately you've put the solution together, so the right temperatures according to the structure of DNA, and in that we mean the CG and the AT pairings. So as we can see, the PCR amplification process is quite complex, and we need to put the correct input into the PCR machines in order to yield the final results that we want. But another question might be, why use PCR? Well, PCR has actually found its way into many different fields of research, one of those being forensic investigations. So if we find a very small amount of genetic material at a crime scene and we need to cross-reference that in order to find out who shot the sheriff, so to speak, we might take this small sample into a lab, use the PCR method and yield a greater volume of this code in order to use that as a sample. We also might use it for genetically modified organisms. So let's take food, for example, because everyone's crazy about GMOs these days. We might find something desirable in a particular species of fruit or vegetable. For example, it's a gene responsible for converting starch into a monosaccharide, so it could actually change the taste. It might make something a little bit sweeter. So we might actually use PCR to extract that part of a DNA sequence in order to replicate it and yield sweeter fruits and vegetables in the future. And an obvious field is medical research. We might use this to find out if someone has a predisposition to a disease, so there might be a mutation in the genetic sequence, or there might be a particular mutation of that sequence which predisposes them to diseases or conditions.
The polymerase chain reaction is a fantastic method in diagnosing infectious diseases. However, currently the polymerase chain reaction and other molecular diagnostic methods can take days to weeks to confirm a diagnosis. The future will see the creation of rapid, sensitive diagnostic tests in infectious diseases that allow clinicians to make sound judgments in real time. This means advancements in nucleic acid amplification technology of the PCR reaction to make future diagnostic tests faster and less expensive, so that clinicians can make more rapid diagnosis and directed therapy a reality. A future advancement in PCR-based diagnostics is real-time detection of the polymerase chain reaction products. Real-time detection methods can be combined with miniaturized rapid PCR technologies. This can design a microchip or microcapillary apparatus that can amplify, detect, and characterize a microbio DNA target within minutes. The polymerase chain reaction have been miniaturized so that huge amounts of a sample are produced in only a few minutes. Using a micro device, 20 cycles could be performed in as little as 90 seconds. The advantages of small volume PCR reactions include reduced costs from the use of fewer reagents and gives the ability to analyze small fragments of a sample so that multiple tests can be performed on a small amount of tissue or fluid. Extensions of the polymerase reaction technique we will see in the future is panels of the polymerase chain reaction assays that are developed which will target microbes involved in specific syndromes such as pneumonia, meningitis, and diarrhea. Rather than having to grow an intact microbe, one will be able to grow a segment of its DNA, replacing culture media with the PCR reaction mix and the incubator with the thermal cycler. Another future application of the technique is an in vitro application of both upstream and downstream regions, called the inverse polymerase chain reaction. As a major limitation of PCR is that it enables amplification of only one region of the DNA situated between the two primers. Using conventional PCR procedure, DNA sequences that lie immediately outside these primers are inaccessible. The inverse polymerase chain reaction can be used to walk outside a region unknown. This allows clinicians to analyze the region adjacent to a specific sequence and proceed along a stretch of uncharacterized DNA. The inverse reaction would have many useful applications in genetics, more specifically to rapidly obtain flanking regions of unknown sequences in order to more rapidly diagnose diseases. As well as the future seeing more rapid PCR diagnostic tests, we may be able to use the polymerase chain reaction to answer questions such as, how long does a microbio disease persist in tissues after the antibiotic treatment? Does microbio DNA persist in tissues after organisms are gone? Does microbio persist in the blood? Can the polymerase chain reaction be used to predict the relapse or persistence after initiation of effective treatment? How can the polymerase chain reaction distinguish between colonization, latent infection, active infection, and relapsing infection? Clinicians will need to compare polymerase chain reaction-based microbio detection methods with existing diagnostic methods to answer these questions to create future experiences and evidence.
Polymerase chain reaction, or PCR, is one of the most advanced biochemistry techniques of the present days. The reason PCR is so successful is the many advantages it has. These advantages are sensitivity, specificity, and speed. One of the biggest advantages of PCR is that it's very sensitive. The DNA of interest can be amplified with the DNA from just one cell. Therefore, very small amount of starting material is required. Also, old and degraded DNA very often yield enough starting material to amplify the DNA of interest. DNA samples as old as 40,000 years can be used for replication. Experts have used PCR to clone DNA fragments from the mummified remains of humans and extinct animals. In addition to sensitivity, PCR offers the potential of remarkable specificity. Specificity in PCR derives from the fact that each distinct microbe has unique DNA. One can design oligonucleotide primers complementary to conserved regions of DNA so as to detect more diverse groups of organisms. Another big advantage of PCR is speed. PCR can be completed in minutes to hours, and so can the identity confirmation process. After 20 cycles, the DNA segments have been amplified more than 1,000,000-fold. After 30 cycles of polymerase chain reaction, the amount of the original DNA sample is increased to 1,000,000,000-fold. Therefore, a turnaround time of only 1 day is not too unrealistic for many types of assay. Although polymerase chain reaction has a lot of advantages, it carries a few limitations as well. Its limitations are its high risk of contamination, false positive reactions, and its costs. As said before, sensitivity is the biggest advantage of the technique, but at the same time, it's its major limitation. Increased sensitivity is obtained because the target is enriched in the first round of PCR. The problem with PCR is that it's highly prone to contamination with amplification products, and thus must be performed with extreme care and interpreted with even greater caution. The usual effort to inactivate amplification products in order to prevent contamination do not work in PCR, because one needs to use amplifiable templates from the first round, in round 2. Opening PCR reaction tubes after round 1, transferring amplification products to new tubes are conducive of contamination. Contamination can originate from 3 different sources: DNA from other test samples, DNA from experimental materials such as recombinant clones, or DNA generated from previous amplification from the same target sequence. The last source of contamination, often called carryover contamination, has proven to be the most troublesome. Ironically, false positives reaction are the Achilles heel of PCR. False positives occur because PCR may amplify contaminating DNA that finds its way into a sample, even when that DNA is present in very, very small amounts. DNA contaminates sample through several means. The most important means of contamination is through amplification product carryover. A single PCR can generate billions of DNA copies, each of which is capable of acting as the target for future PCR reaction. If even a submicroscopic portion of a positive amplification reaction gets into the environment where subsequent PCR reaction is mixed, the false positive reaction may occur. PCR reagents, pens, tubes, tube racks, hands, and door handles, almost any object really, are capable of harboring or transmitting PCR amplification products. To avoid this, usually, laboratories are divided into pre-PCR and post-PCR rooms. Materials are not allowed in the PCR room unless are new or they have been decontaminated. The costs involved in PCR are also an issue. This technique is expensive. The cost of PCR reagents and equipment is substantial. The requirement of pre- and post-PCR laboratory area means that the process involves a fair share of laboratory space. Furthermore, there is a cost associated with extensive training required to teach the microbiologist how to perform the assays.